Hello, welcome back to my channel. For today, my group would like to present our case study for BIO270. Before I start presenting about the introduction, I would like to introduce my group members first. So, my group consists of 4 members including myself. These are all the faces and names. And below here is our student ID. So, let's proceed to the content of this video. Our group has chosen case study number 2. The question of our case study is, how do scientists create fake blood? When it is compared to the genuine things, how effective is it? Before we go further, I would like to share a little bit about blood. Here is some history of blood. This is Karl Steiner, or known as the father of blood grouping and immunochemistry. He is popular because he is the first person that discovered blood. Next is William Harvey. William Harvey is an English physician. In 1628, he was the first one to discover how blood circulated around the body, with the heart pumping blood into the body through arteries and the blood returning back to the heart through the veins. Next, let's talk about why blood is red. Red blood cell, or known as RBC, contains hemoglobin, which means a protein that carries oxygen. Blood gets its bright red color when hemoglobin picks up oxygen in the lungs. As the blood travels through the body, the hemoglobin releases oxygen to the different body parts. Meanwhile, for the shape of blood, RBC are shaped kind of like donut that didn't quite get their whole form. It shapes like by concave disc, a shape that allows them to squeeze through small capillaries. It is also provide a high surface area to volume ratio, allowing gases to diffuse effectively in and out of them. Next, Farahia will continue the presentation.
In 1616, William Harvey described how blood is circulated throughout the body. In the years to follow, medical practitioners tried numerous substances such as beer, urine, milk, plant resins, and sheep blood as a substitute for blood. The first successful human blood transfusion were done in 1667. Unfortunately, the practice was halted because patients who received subsequent transfusion died. In 1854, patients were injected with milk to treat Asiatic cholera. Physicians believed that the milk helped regenerate white blood cells. However, it was soon discarded and forgotten as a blood replacement. In experiments done on frogs, scientists found that they could keep frogs alive for some time if they removed all their blood and replaced it with a saline solution. After much research, saline was developed as a plasma volume expander. Other materials that were tried during the 1800s including hemoglobin and animal plasma. In 1868, researchers found that solution containing hemoglobin isolated from the red blood cells could be used as blood replacements. In 1871, they also examined the use of animal plasma and blood as a substitute for human blood. First, scientists found it is difficult to isolate a large volume of hemoglobin. Second, animal products contain many materials that were toxic to humans. Removing these toxins was a challenge during the 19th century. Karl Steiner is the father of immunochemistry. Using blood samples from his colleagues, he separated the blood cells from its serums and suspended the red blood cells in a saline solution. He then mixed each individual serum with a sample from every cell suspension. Clotting occurred in some cases, in others there was no clotting. Steiner determined that human beings could be separated into blood groups according to the capacity of the red blood cells to clot in the presence of different serums. He named his blood classification groups A, B, and O. A fourth group, which is AB, was discovered the following year. The result of this work was that patient and donor could be blood typed beforehand, making blood transfusion a safe and routine medical practice. This discovery ultimately earned Landsteiner the 1930 Nobel Prize in Physiology or Medicine. During World War I, a gum saline solution containing galactosogluconic acid was used to extend plasma. In the 1920s, studies suggested that this gum solution had some negative health effects. By the 1930s, the use of this material had significantly diminished. World War II reignited an interest in the research of blood and blood substitutes. Plasma donated from humans was commonly used to replace blood and to save soldiers from hemorrhagic shock. Eventually, this led to the establishment of blood banks by the American Red Cross in 1947. In 1966, experiments with mice suggested a new type of blood substitutes, which is called perfluorochemicals (PFC). This gave scientists the idea to use PFC as a blood thinner. In 1968, the idea was tested on rats. The rats' blood was completely removed and replaced with a PFC emulsion. The animal lived for a few hours and recovered fully after their blood was replaced. This prompted some researchers to begin looking for hemoglobin solutions and other synthetic oxygen carriers. Research in this area was further fueled in 1986 when it was discovered that HIV and hepatitis could be transmitted via blood transfusion.
Okay, now I'm going to talk about how effective is artificial blood compared with the real things. Okay, first one is the definition for blood. Blood is a vital, life-sustaining fluid that picks up oxygen in the lung and then carried it to the heart and the rest of the body. Blood performs many functions such as transporting nutrition from the digestive system, removing toxin and waste, and fighting germs. Blood is composed of a watery substance called plasma as well as three different types of cells or part of cells that float in the plasma. The form elements are red blood cells, white blood cells, and platelet. Next one is the artificial blood. Artificial blood is a substance used to mimic and function blood. Although it contains no blood components such as red blood cells, white blood cells, platelets, or plasma, it removes carbon dioxide and transfers oxygen throughout the body until bone marrow can produce more red blood cells. Okay, for the third one is why is artificial blood is needed. If a patient loses too much blood during traumatic injury or a surgical operation, he may need a blood transfusion. Despite the nation's numerous blood banks, there is a critical shortage of human blood available today for medical purposes. For example, nearly 33% of Americans will need a life-saving blood transfusion at some point in their lifetime. With 4.5 million Americans receiving blood transfusion each year, the number of patients requiring blood is now outpacing the number of blood donors. Okay, so these are the main categories of artificial blood. The first one is perfluorocarbon or PFCs. PFCs are synthetic artificial blood products derived from fluorine and carbon containing chemicals. They are chemically inert but more effective than water or blood plasma in dissolving and absorbing oxygen in the lungs and then transporting oxygen throughout the body. Unlike the red-colored HBOCs, PFCs are usually white. However, since they do not mix with blood, they must be emulsified before they can be given to the patients. PFCs such as good oxygen carrier that researchers are now trying to find out if they can reduce swollen brain tissues in traumatic brain injury. PFCs particle may cause flu-like symptoms in some patients when they exhale this compound. Okay, this is the example for PFCs, which is oxygen, developed by Alliance Pharmaceutical Corporation in San Diego. It's a PFC-based oxygen carrier currently approved for phase second trials in both Europe and United States. Oxygen initially showed a promise for decreasing the need for donated blood during surgery. However, phase 3 trials were stopped recently because patients receiving oxygen show a higher risk of stroke compared to control receiving donor blood. Okay, for the second one is hemoglobin-based oxygen carrier, which is HBOCs. HbOCs are manufactured from sterilized hemoglobin and look somewhat like a real blood. These dark red colored blood substitutes are often made from RBCs of expired human blood, cow blood, hemoglobin producing genetically modified bacteria, or human placentas. The artificial hemoglobin molecules are modified to create a sturdy structure and to function without protective cover of RBCs. HbOCs are smaller than natural RBCs, while natural RBCs remain in the bloodstream for about 100 days, but HbOCs circulate in human blood only for a day. Side effects of HbOCs may include elevated blood pressure, abdominal discomfort, and a temporary reddish coloration of the eyes or skin. Okay, so this is the example for HbOCs, which is Hemopure. Hemopure was sponsored first by BIO270 Corporation and later by OPK Biotech. It is currently used in the hospital in South Africa. This oxygen therapeutic drug was approved by the government of South Africa because of the country-wide spread HIV contamination of the blood supply. However, it recently has been targeted for removal from the market in South Africa. Hemopure is made from chemically stabilized, cross-linked cow hemoglobin using simple and less expensive biotechnology than required for other HbOC blood substitutes. Okay, so this is the comparison for RBCs and artificial blood. The first one is artificial blood is faster and better oxygen distribution because this molecule allow full capacity oxygen transport immediately after transfusion, while RBC transfusion usually starts slowly and stored blood takes longer to reach full capacity of oxygen. The second one is longer shelf life. For artificial, it can be stored at room temperature for prolonged period 1 to 3 years, while RBCs it can be stored for around 35 to 42 days and be kept refrigerated until used. Okay, for the universal compatibility, since all the protein components are removed, the human immune system does not recognize it as a foreign entity. Hence, the necessity of compatibility testing based on blood group is avoided. The possibility of clerical errors which might result in mismatched transfusion is also avoided. For RBCs, type O negative blood is the only blood type that is safe to use. The reason is that it is compatible with all blood types. Type O negative is known as the universal blood type. Okay, the last one is for transmission of infectious. For the artificial blood, products are sterilized and hence chances of viral or disease transmission are elevated. For the RBCs, HIV, hepatitis B, hepatitis C are three of the most common bloodborne pathogens.
Next, we are going to see the conclusion. There are many advantages to using blood substitutes over regular blood, such as the robustness, non-refrigeration requirements, and capability to be used in the pre-hospital settings. How effective is it compared to the real things? The purpose of the blood is to replace RBC. Unlike real blood, artificial blood serves only one purpose, that is to transport oxygen and carbon dioxide throughout the body. There are many functions for blood, and artificial blood only fulfills only one of those functions. So in addition to carrying the oxygen and carbon dioxide, blood performs other essential functions such as coagulation and immune defense. Their chemical inertness makes them more effective than water, and blood plasma are dissolving and absorbing oxygen in the lung and transporting it throughout the bodies. And this is our exam references for the artificial bloods. That's all from us. Thank you.